Hello, my name is Justin Archer, and this is my educational presentation on protein structure and diversity. Um, the intended audience for this presentation is an introductory undergraduate biology course, and just provides a brief overview of the topic, protein structure and diversity. Well, today's presentation is on protein structure and diversity, so let's go ahead and jump in. First off, what is a protein? Well, we're pretty used to proteins in our daily lives. We eat them all the time. We need them. Our bodies need them as a dietary um, requirement. We find them in our meats and our poultry, our fish, beans, peanuts, um, many different foods that we can get protein from, and it's vital, like I mentioned before, and we'll talk about that some more in a little bit. These are molecules, okay? Proteins are molecules. There's really three things that I want to bring out here, and in this presentation. One is the fact that they are molecules. They are small. They're made up of a conglomeration of atoms. They are also machines. These are cellular machines. They do the work inside of cells. Just about any cellular process you can think of has proteins behind it. And there's a multitude of proteins as well. There is a multitude, many different kinds, many different shapes, many different functions, and we're going to explore these three facets of what proteins are. They're molecules, they're machines, and there is a multitude of them. So why is understanding proteins important? Why should we even study them? Well, they're the main building blocks of the human body. Like I said, any cellular process that you can think of is going to have proteins behind it at some point. When we study them, they can tell us how food and drugs are processed, giving us vital information to develop new drugs and develop healthier diets as well. They tell us how cellular functions are carried out. So how cell growth and differentiation and division are carried out. That's all mitigated by proteins. Okay, and so when we study these proteins, we're studying the functions of the cell ultimately. And then lastly, they can cause disease directly and indirectly, both. And so the fact that they can be the source of diseases means that we should study them as well. So they're extremely important, extremely, extremely important, and they're incredibly designed, as we'll see here shortly. So this is why we want to study them, because they tell us a lot of information about our bodies, and about how we can overcome the diseases that affect our bodies. So speaking of some diseases, there's diseases that are caused by protein deficiencies, where we are not getting enough protein in our diet, or enough certain specific proteins in our diet, and these can lead to diseases. These are pretty common in third world countries, as you might imagine. There's also protein misfolding diseases. So this is where, as we're going to talk about in a second, proteins fold into their 3D shapes, and when they fold wrong, they can actually cause diseases, because um, their shape really is what determines how they function. And so when they're folded wrong, they function improperly. An example of this is sickle cell anemia. So sickle cell anemia is a disease that's actually caused by a mutation in a gene that codes for hemoglobin. That's the protein in red blood cells that binds and carries oxygen throughout your body. The mutation in the gene actually results in a misfolding of this protein, and that causes the entire cell to take on this sickle shape, right here. And this can actually lead to clogs in the blood vessels. It also can't hold oxygen as well as the normal form of hemoglobin, which causes the cells to look normal. There's also these things called prions. Prions are proteins that they actually are infectious, so they cause regularly folded proteins to fold wrong, to actually misfold, and they induce that misfolding on these other proteins. These protein disease or these prion diseases actually can be inherited sometimes, depending on the disease, the specific disease. They can be inherited. They can be um, they can be contagious, so they can actually be passed on if somebody comes in contact with infected tissue. Some prion diseases actually include Creutzfeldt-Jakob disease, Kuru, fatal insomnia, and variably protease sensitivity prionopathy. And these are just a few different diseases. Ultimately, a lot of the time, these prion diseases affect the brain, and they cause a conglomeration of proteins in the brain, which can interfere with regular brain activity. They are rare. These diseases are rare, but when somebody has them, they're extremely serious and need to be taken very seriously. So some other things too is that venoms and poisons are proteins. Not all the time, but a lot of the time. Venoms and poisons are actually proteins, specifically venoms, and of course, the little side note, the difference between a venom and a poison is that a poison just needs to come in contact with someone. Think of like poison ivy or something, whereas a venom has to actually be injected. So like a snake bite or a tick bite or something like that. Not ticks necessarily, but like spiders and things like that. When they bite you and inject venom, they're injecting a protein that will wreak havoc on your body. Some examples of this are actually the conopeptides. These are found in cone snails. So cone snails, it's a type of mollusk. They're venomous mollusks that hunt fish, worms, or other mollusks, depending on the species. The venom from these cone snails actually, they contain conopeptides. Conopeptides are proteins. They actually inhibit various receptors and channels in order to paralyze their prey. So um, their nervous system is no longer able to function properly, and then they're able to envelop and consume their prey as well. Some species have venom that's actually potent enough to even endanger human life, and so it's very, you got to be careful when you're poking around in rocks and stuff that are out on the beach, if you're in an area that contains these animals. Abrin is a poison. It's a um, it's a poison that's found in rosary pea seeds, and it's highly toxic. Um, and Abrin actually it's a protein. It actually inhibits ribosome function. Ribosomes are what create proteins, what actually make the proteins. And so this actually causes the cessation of protein formation and eventually, of course, death if the dosage is high enough. Because if your body can't make proteins, it can't survive for very long. So in talking about proteins being a molecule, we got to talk about their structure a little bit. So proteins are made up of amino acids. Amino acids are a group of atoms that have come together to form a molecule. There's a central carbon atom here with a hydrogen attached to it. That's in every single amino acid. And then also in every amino acid, there's this carboxyl group over here and an amino group over here on the left. That central carbon atom, that hydrogen, the carboxyl, and the amino group are in every single amino acid. What varies between the amino acids is actually the side chains, which is represented here by the letter R. And there's actually 20 different kinds of amino acids that are used in the human body. You can see all of these have the carboxyl group and the amino group. how they vary are in these R groups over here. Um, peptide bonds actually link them. So multiple amino acids will link up between let's go back here to this picture. They'll actually link up. The amino group of one will attach to the carboxyl group of another, and that actually forms a peptide bond, and those will string together in a long chain with their side chains coming off here. You can see there's a side chain coming off here and a side chain here of the different varying amino acids. Each amino acid has its own properties because it's its own combination of atoms, right? And so there's some that are non-polar. These ones are non-polar, and these ones are also non-polar. These ones are aromatic compounds, so or aromatic R groups. But you've got non-polar, which might they don't like water, right? Water is polar, like things that are like them. So these non-polar R groups are not going to like water, whereas the polar R groups are going to like water. You've also got positively charged R groups and negatively charged R groups, and so these actually determine the behaviors of the proteins. So you have a sequence of these proteins, right? Or you have a sequence of these amino acids, which we can see here. So this is representing an entire protein that is folded, and this linear, this line that is going through is actually the amino acids all strung together. They have their R groups facing in and out and whatnot. And there's actually 3 non-covalent bonds that help fold these proteins and help them to maintain their shape. So you have the electrostatic attractions. So when you have R groups, one R group that's negative and another R group that's positive, they attract each other, and so they're going to want to be close to each other. You've got hydrogen bonding. When you have an electronegative oxygen that is near a hydrogen, they're going to be attracted to each other. That's going to help stabilize a protein structure, help it fold in on itself. You're also going to find van der Waals attractions, which are just the random variations of the electrons in the atoms. There's more on one side, which makes it more negative on that side for just a half a second, not even half a second, like fraction of a millisecond. But when that happens enough, they have very weak interactions with each other, right? And so these 3 interactions are very, very important in terms of how proteins fold and determining that they'll fold properly. So the sequence of these amino acids, what's next to what, determines how they're going to how the whole chain is going to be pulled together and folded in on itself. There's actually 4 levels of structure to a protein. So the first level is the primary structure, which is just a sequence of amino acids. It's a linear sequence of these amino acids. Then we have the secondary structure, which is going to be kind of the first layer of folding, if you will. There's a few different structures that come along with a secondary structure, such as the alpha helix. So we find these over and over and over again in different proteins, kind of a spiraling shape of amino acids where, and it's all held together by hydrogen bonds, as you can see here. And you have these R groups that are facing the outside of this spiral shape. And oftentimes that's how it's represented, is as a spiraled ribbon. You also have beta Pleiadid sheets. These are kind of zigzag sheets of amino acids that are also held together by hydrogen bonds, but they kind of zigzag back and forth. One goes in this direction, another strand goes in that direction, and another strand goes in that direction, and they're all attracted to each other. We represent it a lot of times as these arrows that are pointing in different directions. We see these all over the place in different proteins as well. And so both the alpha helix and the beta pleated sheets form the basis of the secondary structure. So you have primary structure, which is the linear form, secondary structure, which is the first level of folding. Then you have tertiary structure, which is where all of these secondary structures fold in on themselves. So you've got these alpha helices here that are in the spiraled ribbons. Then you also have the beta pleated sheets, which you can see here as the flat ribbons. And they all will fold together. Sometimes you'll have only alpha helices in the structure, sometimes only beta pleated sheets, such as in beta barrels and stuff, but a lot of times you'll see both in the same protein. And so this is what's needed for a protein. Primary, secondary, and tertiary structure, that is a protein in and of itself. However, sometimes multiple proteins can come together and join together through intermolecular attraction and form quaternary structure. So hemoglobin, which we were talking about before, is an example of this. And each of these colors is actually a different protein subunit. Each subunit has a tertiary structure. When they come together, they form a quaternary structure. So those are the 4 levels of protein structure. So next, protein is a machine, okay? Proteins, they do things. They don't just have things done to them, such as like maybe carbohydrates. A lot of times, I mean, carbohydrates do things as well, but a lot of times when we're learning about them, they're used in, we break them down for energy. Or DNA has things done to it. It's red, right? But it doesn't really do anything on its own, not normally anyway. But proteins, they do things. They are, like I said before, they are the workhorses of the cell. So a few different kinds of proteins, right? We've got motor proteins. These guys actually literally, these ones specifically, kinesin, actually walks along these microtubules. It's really incredible. And through just different chemical reactions, the head down here actually puts almost like one quote unquote foot in front of the other, and it walks along these microtubules carrying vesicles and transporting different molecules around the cell. You've also got transport proteins, which are in the membranes of the cells. They allow certain substances in without allowing other ones in. So a lot of substances can't pass through the cell membrane. Water, for example, has a really hard time passing through the cell membrane by itself. So there's actually these proteins called channel proteins. It's a type of transport protein. This one's called an aquaporin. It actually allows the water to go through that channel, like a little bridge that allows it to pass through the hydrophobic interior of the cell membrane. We've also got enzymes. This is a huge group of proteins, and these are the real workhorses, if you will, of the cell, because they catalyze a lot of reactions. So they help reactions that would normally take place at a slow rate. They help them to go faster, and a lot of reactions that would take years and years to actually happen can happen in just a fraction of a second with enzymes. So with enzymes, they have an active site here, which has which is just perfectly shaped for a substrate. And the substrate could be a sugar that comes in, attaches to the active site. The enzyme does its thing. Maybe it takes an ATP or something. It uses some energy to break apart the substrate, and usually it's in a hydrolysis or a um, dehydration reaction, and it'll break it apart, or sometimes it'll actually build things. So it'll take this process happens in reverse, right? Where you have the enzyme, the substrate, the enzyme substrate complex, and then the enzyme with the products. Well, sometimes you'll have 2 substrates that come in and are bound by the enzyme. The lock and key model is a model for how enzymes work. It basically says that the substrate fits perfectly, like a glove or like a key in a lock, to the active site. So enzymes are highly specific. They react generally only to what they're supposed to react with, because the active sites are so specific in their shape. And this is really where the idea of shape equals function comes into play. The shape of a protein equals how it functions in the cell. If it's shaped wrong, it will function wrong. If it's not functioning properly, it's probably shaped wrong. They are highly, highly intertwined concepts. Um, that is an important concept to understand when trying to understand how enzymes work. There's a lot of different kinds of proteins, and here you can see a graphic of some drawings of different kinds of proteins. A lot of these are, well, some of these are enzymes. The way you can tell it's an enzyme, so like catalase is an enzyme because it has that ASE um, uh, at the end. Alcohol dehydrogenase, aspartate transcarbamylase. These are going to be enzymes, and they're going to be catalyzing certain reactions. So there's motor proteins, which we talked about before, that transport things throughout the cell. There's transport proteins, so the motor proteins actually carry things, right? The transport proteins are going to be the ones that allow certain things in the cell and other things not to come into the cell. Enzymes are those that catalyze reactions. You have chaperonins, which are a really fascinating group of proteins that will take the linear strand of a protein, so the primary structure of a protein, and it will put it in a very specific environment so that that protein can fold just perfectly, and then it will release it. Whereas if it didn't go into that little capsule of a chaperonin, it would fold wrong. It's very fascinating. There's signal proteins that, such as certain hormones, right, that will attach to receptors on the surface of cells or on the surface of membranes, and will start a cascade of events that signal maybe the transcription of a certain gene or something like that. There's structural proteins like collagen. Actin is another one that provides structure. Microtubules, they provide structure to the cell. And there's filamentous proteins. So these are filamentous and globular proteins, refer more to the shape. So filamentous might be like the collagen or the actin. Globular are going to be proteins that are much more, well, globular in shape. And so the last thing I really wanted to talk about is ways that we can learn more about these proteins. And there's actually a really cool tool called um, uh, uh, chimera. It's a protein modeling software, or viewing software, I guess you could say, where you can actually take a certain code, put it into the program, and you can see an animated protein will pop up, and you can interact with it. You can look around. You can do different things with it. So I'm going to show you here real quick. So all you have to do is you just type in the protein that you're looking for. So like hemoglobin, and then you put PDB next to it. And you'll find this number, PDB stands for Protein Data Bank. You find that code, you put it into the program, which is UCSF Chimera. You could do a Google search for it. You put that code into the PDB area right up there, and click fetch, and it brings you the protein that you want. So if this is hemoglobin. And you can turn it around, look at every little bit. You can zoom in, zoom out. There's a lot of things that you can do with this program. And you can look up videos, honestly, on how to use it. So here you can actually label the different subunits as different colors. And you can see them stand out even more. And if you hover over certain parts of the structure, you can see the individual amino acids, like the alanine and serine, that's in the protein. You can do other animations to show the surface. So this is kind of similar to what it might look like if we could actually see the protein. This is kind of what it might look like with all of the individual atoms that are there in the space filling model. You can also look at the hydrophobicity of the surface, looking at where water is attracted, where water is repelled on the surface. You can look at every individual atom if you'd like. There's other things you can do with it too. You can measure the distances between atoms. You can measure uh, you can just determine a lot of data. You can look at different conformations, right? So like with hemoglobin, whether oxygen is attached or oxygen is detached, the shape is going to be a little bit different depending on the two. And so you can pull up those two actually in the same window and compare the shapes if you wanted. There's all sorts of things. It's a great resource for students and for um, teachers and for researchers who are just wanting to be able to see these a little bit better, or need a picture of a certain protein. So, and then these are my references that I used for the presentation. I hope you enjoyed the presentation. It is a very fascinating topic, and lots and lots of painstaking research has gone into the field of proteomics, which is the study of proteins. And it's going to be very interesting to see where science takes us as more and more research is done. Um, Thank you for your time, and I hope you enjoyed.